So far in this chapter, we've talked about gene expression, where we take a gene and we go from DNA to RNA to a protein to a trait. Going from DNA to RNA, that's called transcription. Going from RNA to protein, that's translation. The central dogma of this is this process. So looking at a eukaryotic cell, this is the nucleus. Right here we have double-stranded DNA up here. Another way to put it, where does transcription occur? Within the nucleus, and it yields pre-mRNA. And then we go through RNA processing to get mRNA. The messenger RNA goes outside into the cytoplasm for translation to occur, which involves a ribosome and a few other components to make the final polypeptide or the functional protein. Okay, a couple differences between RNA. It's a ribose sugar, whereas in DNA it's a deoxyribose sugar. Notice it is single-stranded and DNA is double-stranded. The nitrogenous base pairs are a little different as well. Uracil instead of thymine for RNA, and U pairs with A and C pairs with G, whereas in DNA, A pairs with T and C pairs with G. And it's single-stranded. Lots of different types of RNA. The ones we're going to be concerned with today are mRNA, tRNA, and rRNA. Okay? So right now we're going from DNA to RNA. Again, the process is called transcription. So let's take a look at transcription, various stages. We have initiation, elongation, and termination. Taking a look at initiation, on the DNA, the double-stranded DNA, we have this transcription unit from here to here, also called a gene. And then we have RNA polymerase. ACE tells you it's a protein enzyme binding to the promoter region, which has the TATA box in it. From here, it begins to transcript RNA, okay? It unwinds it and starts to elongate it, and then it'll hit a termination signal and it'll pop off. And now we have this completed RNA transcript. Now let's take a look right here at what's going on. So we have DNA or RNA polymerase, and it opens up, unwinds the DNA, breaks the hydrogen bonds between it, and we have a template strand, okay, which is what we're coding for. We get this, RNA nucleotides drop in, and they attach here, and then just keeps on. Again, we're going from 5' to 3'. Okay, then we're left with this, pre-mRNA. A couple things we need to happen. During processing, we need to have a 5' cap to it. We also need to cut out the introns. Those are uncoded junk regions, okay? So in eukaryotic genomes, we have exons, that's the real gene, it gets expressed. And introns, that's the junk that we don't really care about, and that's the in-between sequences, okay? This is the overall look of it. And here we have RNA polymerase going through with RNA transcript, okay? We have the pre-mRNA, we processed it, we send it out through a nuclear pore, and then the ribosomal subunits come and attach to it and begin reading, translating this. We have tRNA, some are carrying amino acids into the A site and getting added to this growing polypeptide, okay? And all these little shapes are the various amino acids. We know we have 20. For building a polypeptide, initiation brings together messenger RNA, the ribosomal subunits, there's two, a large subunit, and then a small subunit, and then the initiator tRNA, okay, which is this, okay? And now notice that this 3 little letters, here's the codon, and on the bottom of the tRNA, we have the anticodon, okay? And the amino acid is attached to the end of it. Elongation, what we do is add amino acids based on the codon sequence, okay? And then termination, there's an end codon, and we learned that in class, it was stop, okay? That's what it codes for. And can you tell this story tomorrow? That's what we need to know. Okay, we have DNA. We have RNA polymerase. we have pre-mRNA. This is the exon. There's the introns that are getting cut out. We have a 5' cap. We have a poly A tail. We have a mature RNA or mRNA, messenger RNA. Okay, these are the large ribosomal subunit, a small ribosomal subunit. They come together on the mRNA and begin to translate the message. Notice that the various sites, the A site, the P site, and the E site. This is where it enters, comes in, and this is where it exits. That's where it gets longer. Polypeptide 5' to 3' is the direction that the ribosome travels. And what brings it in is the tRNA. There's just a free-floating tRNA. These are the various amino acids. This is the enzyme that puts the amino acid on the tRNA. And this is the growing polypeptide chains. Each amino acid is linked with a peptide bond.